down the road. So in a minute, I'm going to start over again. Again, welcome to the 10th annual second virtual New York Harbor School Marine Science Symposium. My name is Mauricio Gonzalez, and I am the director and founder of the Marine Biology Research Program. Carla, if you can please hit that first slide. Go backward. Okay, so at 1:30, we're going to start to hear from our principal and Kate Mumford, our our 12th grader from the class of 2021. And if you can please just, if you have any questions at this point, you can close out the first screen and just join with this screen here. I do want to remind everybody that this is much better if you use earphones. If you unmute yourself and you don't have earphones, we're going to get feedback, and that won't be so good. So please make sure that either you have earphones on or you mute yourself if you're not talking, so we don't get any feedback. If y'all can hear me, give me a thumbs up, please. Okay, Carlo, if you can hear me, if you can head over to the next slide. Okay, Carla, let's get to the next slide, please. Thank you. And Prophet, you're up.
All right. So good afternoon, everyone. Can everybody hear me?
Yes, we can hear you.
Okay, perfect. So welcome to our 10th annual New York Harbor School Marine Science Symposium. I hope everybody is doing well today. There are going to be rooms throughout the presentation named after different coral reefs, and these waiting rooms will serve as momentary lapses while we either prepare or someone gets ready for their presentation. All right, okay. So, of course, with our amazing program throughout our school, each program has a mission statement. For ours, our program seeks to work with inner city and underrepresented youth to attain access to careers in science, technology, engineering, math, and positions of power through real-world environmental restoration projects that make a difference in our communities and individual lives. Okay, so good afternoon, friends, family, scholars, staff, industry, and post-secondary partners. This year's Marine Science Symposium is brought to you by the New York Harbor School, the Marine Biology Research Program, and the Harbor SEAL Civic Science Monitoring Team, our Harbor School after-school program.
It is now my honor to present to you our Senior Project Manager and valedictorian, Kate Mumford.
Hello, my name is Kate Mumford. So good afternoon to students, teachers, and attendees. I'd like to thank you all for coming to our 10th annual Marine Biology Research Program Science Symposium and our second virtual symposium. I am so excited to have all of you here today to showcase our best research projects, to discuss and learn from each other, and to come together as a community that values discovery and research. Over my time in the Marine Biology program and Harbor SEALs, I've learned so much. I've had to push myself further than I ever thought I could, and I've learned how to be a strong leader. But most importantly, I've experienced growth and maturity alongside my talented and wonderful peers in a truly meaningful community. And with that, I'd like to introduce you to our fantastic principal, Jeff.
Hi, everyone. It's great to be here, and thank you so much, Kate, for saying fantastic principal. I really appreciate that, especially coming from you and coming from this program, which I think is fantastic and amazing. The amount of work that you guys do on a regular basis, and then on top of everything else, to put something of this caliber together for everyone that's here today. And I know the hard work it takes to put together a virtual event. I know the hard work it takes to put together any event, but a virtual event is very tough to do. We did graduation last year and prom, and now that you're having your second symposium virtually is amazing, because you could easily just say, you know what, let's just wait till we get back in person. But I applaud Mauricio and the students of the biology research program for putting this all together. And it's just real amazing work each and every year. I think at this time last year, I said that we would be back in person this year. So right there is the difference between theory and science. I should have been listening to the science and scientists at the time, but I'm almost guaranteeing we'll be back in person in September. I'm afraid to say it this time, but I don't want to jinx it. But I'm super excited to celebrate our seniors next week. I'm super excited for the work that you're going to see during this presentation today. And Mauricio and your team, I can't thank you enough for putting in the time and effort into all of this. Kudos to you. Kudos to your students. This is special any way you look at it, and I thank each and every one of you for being here today as well. So thank you all. I'm very proud. Keep up the great work, and I'm excited for what's coming up next.
All right, thank you just so much for those kind words. And again, we appreciate everything you do for the program as well for our school and of course the seniors this year. With every Marine Science Symposium, we do have a theme that we like to incorporate. This year's theme is going to be Janus, which is a Greek god, and he represents the god of beginnings and transitions in Roman mythology. Janus has two faces, one looking in the past and one into the future, and he resides over doors and gates, being the cause of changes and movement in time. The events in our present have had many historical changes, both good and bad. We have reflected on our past and are now looking towards a new future. Of course, with the year we've had, this was really involved in our year, which is why we chose Janus. We want to look towards a better future after a not so great year.
Thank you, Prophet, for reading this year's theme. It is now my pleasure and honor to introduce our alumnus keynote speaker, Andrew Sommer. Andrew Sommer is a 2015 graduate of the MBRP program. After graduation from the New York Harbor School, he attended the State University of New York, SUNY at Oswego, where he earned a degree in biology and chemistry. While at SUNY Oswego, he was introduced to microbiology research and spent 4 years studying how interactions between gut microbiomes influence the health of fruit flies.
Hi, thank you all for inviting me. I'm going to share my screen if that's okay. And could I just get a verbal confirmation that that people can see my screen?
I can see it.
Okay, well,
It's on.
thank you very much. And I do have to apologize for being a little late because I'm on a different time zone and it really didn't dawn on me to connect that. But I'm really glad to be here, and I want to thank everyone for inviting me, and specifically to Luke, who reached out to me. And I thought a lot today about what I want to talk to everyone about, and I decided that I don't want to talk about specifics of research, but rather my path to a biology PhD program. So I was in the marine biology research program, and I graduated in 2015. And like everyone in the program now, I had an independent research project. And specifically, my research project focused on the concept of ecological succession, which is broadly described as the change in biological communities over time. And I was interested mainly in applying this concept to invertebrates or macroinvertebrates growing on porcelain plates that I put in the New York Harbor. And I'm actually really excited because I was looking into the program and I see people are still working, students are still working on this research. And I'm sure if I have more time, Mauricio can tell you a little bit more about the failures and the successes which came in this project. And I think that's something that happens in all research projects. But nonetheless, doing an independent research project through the Marine Biology Research Program taught me a few valuable skills. And these are skills that everyone who's in the program are learning currently. And perhaps the most important one is project management. So this is something that I'm 100% sure that everyone hears, everyone in the program hears about again and again, and the importance of being able to do this. And I'm here to guarantee you that project management is perhaps the most important thing in being a successful researcher and potentially going on to doing a PhD in biology, and something that I do every day. And of course, you're learning the fundamentals of reading and writing scientific papers. And that's incredibly important. I personally taught classes to third-year undergraduate students on how to do these same subjects that that everyone in this program is already learning. And of course, you're also being able to practice your presentation skills. And so these are sort of 4 key skills that everyone in this program is going to graduate with. And they're the same skills that if you're interested in going to a biology PhD program, that you'll need to get better and better at. So from the New York Harbor School, I went on to go to SUNY Oswego, and I earned a degree in biology and chemistry. And I'm often asked by current undergraduate students who are interested in a biology PhD on what the most fundamental and what the most important part of these programs are, or what the most important part to prepare for a biology PhD program. And they're often asking all about the different classes they should take. And my main response to them, and I think that this is the best single piece of advice I can give to anyone interested in doing a biology PhD, is to get involved in research and to start early. Start in your freshman or your sophomore year. And it's going to be a little bit different than the MBRP research program because you may need to ask and seek out opportunities. So when you get to college, you should look for professors doing research that you might find interesting and send them an email. So I was fortunate enough to have been given the opportunity to work in a lab with a professor named Dr. Peter Newell, where we studied interactions between fruit flies and their microbes. And this is where I was really introduced to the field of microbiology. And so microbes are incredibly important to the development, the physiology, and the health of all animals. And specifically, we were interested in better understanding the interactions between all these bacteria. And by interactions, I'm talking about in the ecological sense. So things like antagonistic interactions where 2 bacteria are competing with each other, or interactions where bacteria are cooperating with each other. And if anyone is really interested in this sort of research, please feel free to reach out to me. But I want to focus more on the skills that doing undergraduate research will help you to develop. And these are the same skills that I mentioned that everyone is already learning in the marine biology research program. And so over the course of your undergraduate career, I think what you should be focused on is improving these skills more and more. So going on to take opportunities to present your research. And then again, project management is fundamental, and learning how to really get better at designing and implementing experiments. And this may happen in your home university, but there are a lot of opportunities for undergraduate students to go out and do research in other places as well. So for example, my research took me to Zurich, Switzerland, where I worked at the Federal Institute of Technology, doing research there. And I think the key is to get involved in research early, and then again, to go and seek out opportunities. So from SUNY Oswego, I graduated and I went to the University of Wisconsin-Madison. And I would like to say that for people in this program, when you go to university and you get involved in research, I think that as long as you work on the skills of project management and scientific reading and writing, it is very easy to get into a PhD program, because that's what they're looking for. They're not looking for your, they're looking to find students that are good at doing research. And everyone in this program, I think, everyone in this program practice, and I'm sure you are all good researchers. And so currently at the University of Wisconsin-Madison, I went full circle, and now I'm back to studying this process of ecological succession, except I'm not doing so on macroinvertebrates on porcelain plates, but I'm rather applying it to the concept of disease ecology. And so my research focuses on understanding how pathogenic bacteria living in dairy barns can get into house flies and biting stable flies, and how those bacteria can colonize and persist within these flies and disperse the pathogens to new environments. And these processes are all controlled through ecological succession. And I know I don't have tons of time to speak, and so I would rather have some time to answer questions from anyone or from students, as long as that's possible. And before, I'd like to thank the MBRP research program for really getting me or starting me thinking about research, and then of course the other mentors that I've had, including my undergraduate mentor, Peter Newell, who was really great, as well as Dr. Carrie Kuhn, who is my current mentor. And my email is on the screen for anyone who may be more interested in this topic, but maybe a little shy to talk now. So please feel free to reach out. Thank you very much.
That was great, Andrew. And while we're getting the other presentation back up, I don't know if when you pulled yours up, it might have bumped us out from the original one. So there we go.
If anyone has any questions for Andrew, you can put it in the chat if you would like, or unmute, raise your hand and unmute your mic just so we know that you're speaking. Yeah, that would be the time to do that.
Okay, so we might have time for maybe one question at this point. Although I don't see, I don't see any messages.
Nan raised her hand, sorry.
So go ahead, Nan, and you can ask you can unmute yourself and ask a question.
Hey, Andrew. That was really interesting, and thank you so much for coming to talk to us. And I was just curious about how marine bio here at the Harbor School prepared you to move over to, I guess, more terrestrial bio with flies. When did you make that transition, and why?
I think in biology there are sort of these fundamental unifying principles, and things that you learn in marine biology can directly translate to terrestrial biology. And so for me, that was sort of learning about these concepts of how communities change over time, and understanding those basic principles in an aquatic setting, also to a large degree occurs in terrestrial environments. And I made the switch to terrestrial, I think. I went to a small undergraduate university, and I was interested in the concepts of microbiology, and there weren't many microbiology professors. And so in some way I was funneled into studying flies, but then I realized that I absolutely love studying insects, and so I kept in that way. But I think the most important thing really is being able to have these project management skills and research skills that everyone in the MBRP program is learning, and those are the most translatable to any area of biology or even other areas of science or non-science.
Thank you. That was great.
We may have time for one more question. I also added the sign-in form, especially for the students in the MBRP. If you can please sign in if you haven't. That's how we're taking attendance today. Okay, you all know me. I'm going to jump in. So Andrew, can you talk to us about maybe one failure that you had in the MBRP? Like for folks that might lose their motivation to continue, what kept you going?
Oh, well, I can talk about some of the failures.
You got 2 minutes.
Okay, the porcelain plates. The original design that we had was to take these porcelain plates and have them floating on these little floating docks. But we really didn't, I guess, take into account how strong these waves of the New York Harbor are, and they got swept away to the sea. And that's sort of all the research, all the data that's kind of gone. So it's a little discouraging, but things like this happen, I think, doing scientific research all the time. And I have failures, both big and small, on a regular occurrence. And I think the important thing to do is to learn from those failures. So with the porcelain plates that floated away, we learned that we can't use these floating docks, and we decided to then find a better way to do it and a more direct way to do it and attach them directly.
Yeah, I remember the struggle. I remember the struggle of going down to the Flupsy and making sure y'all didn't fall in the water. At the same time, trying to do some science with like 20 folks on the Flupsy, everybody, moving these heavy instruments and trying to coordinate with so many folks. Okay, so thank you, Andrew. This has been a wonderful. I haven't seen you in, I don't know how long has it been? Maybe 6 years or so. So it's great to see you again, and thank you. All right, at this point, I'm going to ask our MCs to take it away.
Okay, thank you, Mr. Sommer, for that wonderful presentation, and thank you for answering questions. It was really helpful. It is now my pleasure and honor to introduce our keynote speaker, Dr. Kathleen Nolan. Dr. Nolan taught biology at a variety of New York area's colleges before joining St. Francis College, including Yeshiva University, Columbia University, Iona College, and LaGuardia Community College. She teaches general biology, ecology, genetics, and other topics with research focused on environmental biology and aquatic ecosystems.
That's great. Thank you very much for that introduction. Can everybody hear me okay?
Yes, we can hear you.
Okay, great. And Andrew, thank you very much for that very wonderful talk. One thing I was thinking about when you were talking about mistakes was I was working in someone's lab one time and I made some kind of bad mistake. I threw something away I wasn't supposed to, or whatever it was. And the guy said to me, the researcher said to me, well, he says, people that do things make mistakes. So, so if you sit back and are afraid to touch anything or do anything, then you're not going to make mistakes, right? So you have to jump out there and go for it. Anyways, right now I am sitting in, I wanted to tell you to pin me if you would, so that, not that I have this great ego that you need to see me in full living color or anything, but I have something that I want to show you. It's a live animal, okay? So I want you to be able to see it. Now, if you do this next year and it's in person, I can come in and actually bring these to you, or I can come to your school sometime and show these to you, these animals that I'm going to talk to you about. Now, how did I get interested in those animals? Well, I'm from Vermont, and right now I'm in Vermont. That's what's sort of cool about Zoom. And I'm sitting in a chair, and I'm on a porch, and I'm looking at Lake Champlain off there in the distance. And my grandfather built this cabin that I'm in the 1930s. And so I'm a Vermonter through and through. My parents were from here, and my grandparents. And when I was a kid, I used to collect clam shells and algae and snails and things like that. I was a bit of an amateur naturalist, I guess you could say. And this young girl that I ran into, I was about 11 years old, she had these seashells that I'd never seen anything like them before. And it turned out there were scallop shells. I mean, they're pretty common. Some people pronounce it scallops. Some people say scallops. So I'll go with the Maine way, which is they say scallops in Maine. I think they say scallops in New York. So I started collecting seashells, and I got interested in kind of a more marine biology. And as you know, that involves the ocean. And there's no ocean around here. We're a completely landlocked state. We do have Lake Champlain on the west and the Connecticut River on the east. And I didn't visit the ocean until I was over 15. I was around 15 years old when I visited the ocean. So anyways, I decided I wanted to be a marine biologist, and I went to Northeastern University in Boston. And I highly recommend that school because they have what's called a cooperative work study program. And it's a 5-year program, not because you're held back or anything. It's because you get work experience and you work for 6 months and then you go to school for 6 months. And I got to have 2 really good marine biology jobs. One was at Sandy Hook Marine Lab in New Jersey, and that's part of the Gateway National Park Service, as is Jamaica Bay and Floyd Bennett Field, and you may have heard of some of those places or maybe visited them. And then the second job that I had was up in Portland, Maine. So I worked for the National Marine Fisheries Service there, and I worked in the statistics branch, and this is when I was in college, and we were trying to keep track of all of the fish that commercial fishermen catch, like cod and haddock and pollock. And what do we have here? Oh, do we have a lobster? Yes. So I got very interested in lobsters also. And I got to go onto lobster boats, and I found out that they have this tool. It's kind of a metal-shaped tool that they measure the carapace. This is the shell of the lobster. And if it's above a certain size, then they can keep the lobster. And if the lobster is smaller than that size and that little gap, then they have to throw it. They call it throw it away, right? And so the title of this talk is really The Anatomy of a Lobster, because I just think that they're so cool in that they have so many projections and so many uses. Like they have every possible tool imaginable in their body. We have arms and legs. Those are our appendages, right? But a lobster has tons of stuff, right? They've got these antennae, and these are called tactile, and they can feel the bottom because a lobster is benthic and it moves along the bottom. And it has attenules, which are these little short things sticking out. And those are chemosensory. So they can detect odors. They're like our nose, right? And then these are also called, they're in the phylum Arthropoda, right? Jointed. So they have all these different joints, and they have segmentation, which we have segmentation in our vertebrae, which is kind of cool. So you see evolution through the lobster. And you can see that these are flexible and that they can move these. And they have 10 walking legs. And these are called the chelipeds, these big ones. You can imagine why they might have the rubber bands around them, right? And they, one is called the crusher claw, which is this thick one right here. And this is the pincer, which it will grab its prey. And then they can crush it with the crusher claw. And they even have appendages around their mouth. These things are called maxillipeds, and they actually sweep the food into their mouth, and they have little jaws like mandibles. I don't want to get too close to that. And they have sometimes this thing's trying, just grab me with its pincer, okay? And this is these are called uropods. They can help the animal swim through the water. This piece right here is called the telson at the bottom. And then I love the swimmerets. I just think these are the coolest little things, these little feathery-like things, and that's what they use to swim. Now, this happens to be a male. They have a modified swimmeret, and it's a little harder than, I'll show you the female. I have a pot right next to me that they're going into after this talk, by the way. And they're a little bit more feathery on the female right here. And so what the male does is it has a female on its back, and it puts sperm into like a hole, like at the base of the swimmerets, and that's called the spermatheca. And then what happens is when the female lays her eggs, her eggs come out a different opening, and then she fertilizes them with the sperm that have been stored in the spermatheca. So that's like really pretty cool. And let's see what else. They have a very powerful abdominal flexor muscle this helps them move through the water also. And they have what's called the ventral nervous system. Ours is dorsal, right? And so they have nerve cords that go along the side. And their eyes are they're multifaceted, kind of like other arthropods that you may have read about. And they're just, I just think they're the coolest organisms. And of course, they're commercially fished, right? And they're doing well, so you don't have to worry about that. So I am not going to make them endangered when I put them into my pot later on and to make my lobster rolls that I'm going to make later. And they do turn a bright red. I meant to look that up to find out why they do that. Like, because you usually think of lobsters as being red, right? Well, that's when they're cooked. So, but they are doing well in the Gulf of Maine. These happen to be from Canada. And that's it. So I'll just show you the both of them together so you can admire them. As Michael Judge knows, he's seen spiny lobsters. And those are in the Caribbean, and they don't have these big claws. So if you're snorkeling and you run into a spiny lobster, which I've never eaten those, I don't know what those taste like. They look quite different. They have these very long antennae. And just another little kind of fun fact, they're not at all related to the horseshoe crabs, right? They are related to crabs, like say it's a blue crab and shrimp, but the horseshoe crabs are in a totally different group. They're more closely related to spiders and ticks. So that is the conclusion of what I wanted to talk to you about. And if you become a marine biologist or if you become a teacher, you get to kind of do cool things like this and study really neat organisms and find out more about them. Thanks.
Thank you so much, Dr. Nolan. And I wanted to ask you a question. Is it true that lobsters actually urinate through their rostrum, through their head?
I don't know.
Oh, okay. I learned that a long time ago, and I think that I thought that was kind of weird. Yeah. So, well.
Another thing they have that's very cool is they have something called the green gland, which is kind of analogous to a liver, and you can actually eat that, and it actually tastes pretty good. I don't know, I like things like that, so it's very good.
Yeah.
Tasting.
Yeah, and there's a huge lobster shortage for going on right now. I think to get a lobster roll, it's something like $40, or it's absurd.
That much?
Right? Yeah, around here. Yeah, around these necks of the woods here, and probably even more expensive in New York City, actually. Yeah. So we have time for a couple of other questions. So if you want to go ahead and raise your hand and unmute, you can do so. Anything that may capture your attention about, or if you want to learn more about this stuff, go ahead, Cezanne.
Okay, hi. So I just wanted to ask about what you thought about lobster immortality, and how, like, I know that those two lobsters that you have right there aren't gonna live forever, but I know that if like they keep molting and growing and molting with no predators or predation or bacterial infections, that they can live for a pretty long time. And I wondered what you thought about all of that.
Well, it's like anything. It's like my brother's a fisherman. He's always out on this lake and he's always catching fish. And I don't particularly like to do that. I like seining. I do like doing that. I like catching the fish and then putting them back. And but I also like eating the fish that he catches. So it does bother people sometimes when you tell them you're going to kill a lobster. And but usually people don't have any trouble eating them. So So I'm not going to feel too badly about it, I guess. If they were endangered, like say, for instance, I've studied sturgeon before, which is a kind of a prehistoric looking kind of fish, and it has an uneven tail that looks a little bit like a shark. Those are, they grow very slowly, and they don't reproduce until they're around 14 years old. So people catch them, but they catch them before they've reproduced. So that's going to cause something to become endangered if you catch it before it reproduces. So there is a moratorium on fishing for sturgeon in the Hudson River for like 40 years. So something like that, I would not want to eat something like that that was endangered. So.
Yeah, I want to follow up with climate change. How is climate change affecting the lobster population? Are they moving, or is it affecting them?
You know, it's
in that sense.
You mentioned that. Like, I was just, you know, I probably could have read tons and tons more articles in preparation for this, but I did look at a couple, and they said that they are tending to move a little bit more offshore, but not more north or more south or anything. So probably climate change is affecting them because probably the water's a little deeper and maybe it's cooler for them. People don't really know why they're doing that in Maine. So
yeah, and maybe as another follow-up question to that, do you know anything about the policies that are being, like, I know that there's folks that are fighting against the lobster farmers because they trap their implements tend to trap whales, and so I think that you know, what I read was that up in Maine there's this one guy who's fighting really hard, and I think he has like a lawsuit where he might win because for some reason or other, and so do you know anything about that policy sort of the lobster fishing and whaling? Or whale population? I should say whaling.
I find that hard to believe. I mean, I want to look into this now, but I've gone out on lobster boats, and they have like these wooden traps that are about, I don't know, maybe 4 or 5 feet long by 2 feet tall, 3 feet tall. And what they have is they have like a funnel inside of them, so the lobster can get in, but then they can't get out. And when I went out with the lobster fishermen, like I said, they had this tool, and it was mostly just lobsters that were inside that trap. You know, I'm trying to remember. I don't think there was anything else in there. So, I mean, it would have to be a pretty small whale, I think, that would get in there or get trapped. There are other types of gear that is very harmful to the environment. Like, for instance, that was part of my job, was to interview fishermen and find out what kind of gear, they called it gear, what kind of gear they were using. And say, for instance, swordfish, they would go after swordfish, and they'd come back with maybe 200 swordfish. And they said that they put 2,000 hooks on these long lines. And then they'd say they caught just as many sharks, and they would just discard the sharks. So they would kill 200 sharks to get 200 swordfish. That's a little crazy to me. So I know that swordfish are on, like, there's like a list that is kept by, you know, the different departments of conservation of which fish are like kind of friendly to eat and not environmentally. And swordfish is on that list as not being friendly environmentally. It's hard, you know. It's. I remember when I worked for the fisheries, and the fisheries in Maine, like, has really been brought down. Like, for instance, there's very few cod and very few haddock, and I was there like many years ago when I was in college, and they were overfished really. But there's all these different interstate boundaries and regulations, and people have fights, and they'll try to close a fishery, like they closed a shrimp fishery in Gloucester, Massachusetts. And then these guys would come, the fishermen would come, and they'd go and fish anyways, and they'd dump a bucket of shrimp on an official's desk. Like, I remember that thought in my head when I heard that in college, like, why would somebody do that? And, I mean, people do everything, right? So there's fighting for those kinds of things. Say, for instance, the seals, right? Like, seals and whales, walruses, polar bears, they're all marine mammals, and they're all protected by the Marine Mammal Protection Act, which was passed in the 1970s. And then what has happened is the seal population, they used to give people money if they could kill a seal, and they would give them this bounty for killing seals. And because seals eat fish too. So, but with that act, these animals have been saved. But then what has happened is you've heard of great white sharks, you've heard of, fish die-offs and things, and some of it is being blamed on these seals. Like they become more abundant, let's say in Massachusetts and off of Long Island. So it's kind of like, how do you balance nature against human needs and wants and safety, I guess. And I'm thinking leave the seals alone, and if the great white sharks eat them, fine, but then people want to go swimming, so.
Yeah, that's a great point, how it's complex, maybe, and it's so complex that almost, any argument can be made and defended, which makes it very difficult to progress often. But a lot of these issues, I think the great thing is that we are able to discuss these issues, and we have so much information now that we need to be careful with, right? Because there is so much information online that could be misleading. And in today's age where there's a heavy attack on science,
right.
It's yeah.
Just one more thing to mention that I was kind of shocked about was I went to the Queen's Zoo a few years ago, and I was doing this project on animal vocalizations, and I was using like sea lions as my model.
Who are you?
And the males will vocalize much more than the females. And
I'm sorry, Kat. Dr. Nolan, can you unmute yourself, please?
Unmute.
oh, sorry, I don't know how I muted myself.
Remember, you're already off mute.
I see that. I probably want to be on, don't I?
Yeah.
Okay, just as a quick announcement, yeah, if you all can please unmute yourselves and just one speaker at a time. And if we have trouble with that, I'm going to mute all, and then the next speaker will go. Actually, we have presentations after this, and we are a little bit over time. So sorry that we interrupted you, Dr. Knoll. Do you want to finish that idea?
Sure. It was about sea lions, and if you go to the Queen's Zoo or the Bronx Zoo, you'll see that these sea lions, some of them have brands on them, and I saw a YouTube video that was kind of horrifying. And what they do is they actually use like a cattle. They burn a symbol on the side of a sea lion, and because what has happened on the West Coast is that because we don't have sea lions on the East Coast, on the West Coast they in the state of Washington and Oregon, they have salmon fisheries, and the sea lions sometimes eat the salmon, and the salmon are considered to be somewhat endangered. So the fishermen will brand a sea lion, and what happens is sometimes they're allowed to kill them. So there are exceptions for this Marine Mammal Protection Act, and some of them in some of the zoos have been able to pick them up as rescues. So because they repeatedly, once they learn how to get the fish, then they I don't know, it's hard to keep them away from the fisheries, and they're these big animals. So, without killing them, I guess. So, but the National Marine Fisheries Service is allowed to. They call it euthanize a certain number of sea lions every year, and I was so shocked when I heard that.
Yeah.
So I thought that was worse than me eating lobsters.
Yeah. I don't feel as bad. We all eat, and that's a topic for another day about like what sorts of foods we can eat from the ocean. And you're all encouraged to go to the Monterey Aquarium website, where they have lists of what sorts of fish are safe to eat, which ones are not, and they rank them and score them. And so please do that if you get a chance. it is important for us to know it's an extra step, but if you can do that step, that's part of the work that we do, is figuring out how to make good decisions with the data that people are gathering in their research projects to make sure that our marine life, not just animals, but also plants and others are well protected. And as you all know, the harbor has been highly impacted. The New York Harbor has been highly impacted in all sorts of ways. It's not just animals that have got extirpated, that have disappeared, but also plants. And so I'd like to thank Dr. Nolan for taking the time to talk to us and answer our questions and to explore this topic. We appreciate all the work that you have done with us over the years. You have been a very generous sponsor and mentor and advisor to our program over the years as a professional advisory committee member for over 10 years. So, and you've been an integral part of our team. So thank you very much, Dr. Nolan. And I'd like to transition over to the MCs. If you all need to take a brief break, you can do so. And we will begin in about in about 2 minutes. Sorry, we can give you a 2-minute break there if you need to run and do something. And we're going to be transitioning into our scholar presentations. So let's check back in 2 minutes, and so that would put us at 2:17. Okay, for those of you who have hung around, I'd like to just mention that I was very amused to see everyone really lost in Gathertown. I apologize if anyone suffered any extreme anxiety from being lost and running around in our virtual lab. And just so you all know, we did actually do a lot of really great science out of using that tool because it allows folks to break off into groups really quickly, and it also gives you the feeling that you're going somewhere and that you're part and that you're not just stuck at home. Excuse me. And so I was suspicious that it wasn't going to run as smooth as when we have only 16 scholars in there. But that was a little bit of a taste of one of the tools that we used. And I'm sure that you're all appreciating the very structured presentation that we have now in Google Meet and the contrast. So, and obviously education will never be the same again with all these tools. All right, so MCs, if you can please take it away from here. All right, so I'm going to begin. And Carla, are you there? If you can just give me if you can unmute yourself and let me know if you're there.
Yes, I'm here.
Okay, thank you. Maybe our MCs are still getting a glass of water there. So let's move on to the next slide. And
the MCs are here.
All right, great. Take it from here.
Okay, so I hope everybody had a good break and had some water. Once again, thank you, Dr. Nolan, especially for the live specimens. That was really cool to see. We will begin our notable scholar presentations. Just as a guide, we will first be doing our SEALS presentations, which is our after-school program, and then we will move on to our winning presentations from sophomore class through senior.
Hello, this is Team Airbenders, and this is our project, Observations of Air Quality in Neighborhoods Around New York City. The 10th graders leading this project is Jalen Boyce and Anna Sheehan, and the mentors is me, Emily Sakova, and Prophet Davidson. Our advisors were Mauricio Gonzalez and Jim Lodge, and this is a project for Harbor SEALs of 2021. So our topic question is there a difference in air quality index between neighborhoods with lower income than those of higher income? And now, for a long time, environmental classism continues to be prevalent even in the 21st century, with families in poverty being affected by climate change the most. And using our eggs to collect data from around New York City, as well as Barbados and Connecticut, we are being able to identify the correlation between air quality and household income. Now, it's no secret that there is a contrast between neighborhoods with lower income versus those with higher income. Some of these differences may include community resources, education quality, and now because of science, we know that there's a variance in air quality as well. The question is, how big is this difference? Now, with air pollution being linked back to asthma, lung issues, and many other problems, this project is very important to both the science community and the general population of New York City.
Our hypothesis was that the lower income neighborhoods would have a worse air quality index than neighborhoods with higher household incomes. Our first objective is to collect air quality data. Our second one was to draw a correlation between household income and the air quality. Our third observation was to make people aware of the effects of environmental racism, and our fourth one was to promote other scientists to investigate differences between community incomes and how it impacts health. Our materials were, we had 8 eggs, 4 in Brooklyn, 1 in the Bronx, 1 in Manhattan, and 1 in Staten Island. We used computers and Google Docs, Sheets, and Slides.
Now for our procedure, the data collected comes from the Air Quality Egg portal at airqualityegg.com. On the website is found by clicking Menu, Labs, and then My Data. Then select the specific eggs data by selecting a time frame, then Refresh Data. The graphs that appear are customizable, and it shows what type of graph is used, what values are being measured, and the egg name. From here, the air quality index values are recorded into a separate Google Sheet for each egg station. Now our project is an observational design, and our proposed experimental is air quality in New York City neighborhoods, as well as our independent variable being air quality index, and our dependent variable being neighborhoods in New York City. Our proposed controls is egg placement, most preferably inside, and proposed constants would be the testing area, which is New York City. And here's our observational design in a chart. So for some safety precautions for the air quality egg itself is to always keep it in a dry environment, away from direct sunlight, and always placed upright, as well as always being plugged into a power source at all times.
We are assuming that the eggs are calibrated so that our data is the most recent, and we can assume that areas that do not have eggs in each barrel have similar data to neighboring areas that have eggs. We can assume that air quality outside is similar to air quality that is in a person's home. And we can assume that the data we collected from March to May is accurate for the whole year. For our limitations, we said that there aren't eggs in every barrel, for example Queens, and that the egg must be plugged in at all times. So if the power goes out or if it is unplugged, the egg will stop collecting data. Some of the eggs are inside and some are outside. Some eggs may not specify whether inside or outside. This is a timeline of our project where we have weekly SEAL sessions and the data of the symposium.
These are our results. Our first figure shows the data we collected for four neighborhoods: Sunset Park, Kensington, Dogan Hills, and Greenwich Village, from March 24th to the May 19th, 2021, and the household income data is from the year of 2019. Our second figure shows those same data points, but with the addition of Morrisina and 2 eggs in Park Slope. These are eggs that are currently running, but not running dependently. This data was also collected from March 24th to May 19th, 2021, and the household income data is also from 2019. There is no direct correlation. Low income and high income neighborhoods breathe the same air according to the data we collected. We have determined many limitations such as little data points and biased station selection. Human and mechanical error when the eggs collected data throughout the time period might have also been a factor. We speculate that the air quality is dispersed evenly through New York City. Similar studies to ours show that high-income neighborhoods in New York City have some of the worst traffic pollution, and it may democratize the air quality.
In conclusion, our hypothesis was not supported by the data collected. Multiple outside influences created bias towards the data, and there was not enough of it data points to fully represent the boroughs of New York City as a whole. Suggestions for future research. We could use a different method of collecting AQI, for example, not an egg because of inconsistent data. Different source of AQI data, policy changes and how they affect the air quality in New York City neighborhoods, how the sources of pollution vary in different neighborhoods in New York City, different variables, not AQI, different range areas, and try to improve the usage of the egg.
Here's our bibliography, and thank you so much for listening. Have a great rest of your day.
All right, so that was one of our SEALS projects. I hope you guys enjoyed. Once again, if you just have any kind of last minute questions about any of the presentations or anything you see throughout the slideshow, you can always add it in the chat, and if there's time, we will definitely get to them at the end.
Hello, this is our presentation, and in it we'll be asking, how have CO2 levels changed throughout the COVID-19 pandemic? By Arlo Kane, Nicholas Bobotoy, Julia Perzella, and Ailish Mullaney. Mentored by Catherine Mumford and Dakota Rogers. Advised by Mauricio Gonzalez of the New York Harbor School and New York Harbor SEALs, and Jim Lodge of the Hudson River Foundation. Before we start, a special thanks to Con Edison and the Hudson River Foundation for supporting our project and research.
COVID-19 has changed the way we will view our society forever. Everyday behaviors have changed drastically, from more frequent hand washing to not being able to eat out at our favorite restaurants. But one of the most profound changes in our lifestyles was the sudden prevention of physical interaction with others. This factor led to an impressively large drop in travel through automobiles. And since cars can produce up to 4.6 metric tons of carbon waste per year, we thought it would be interesting to research the effects of the lockdown on carbon emissions, particularly from cars and other modes of transport.
Our scientific question is, how have CO2 levels changed in New York throughout the COVID-19 pandemic? We have hypothesized that there will be an overall decrease in CO2 levels in New York City during stage 2 of the pandemic, March 2020 to June 2020.
We used 2 separate data locations. One of them was from Dogan Hills in Staten Island, and the other one was from Mauna Loa Observatory in Hawaii.
In the experiment, we posed a scientific problem. How have CO2 levels in New York City changed throughout the pandemic? Our hypothesis is that there will be a decrease in CO2 levels in New York City over the course of the entire COVID-19 pandemic, due to a rapid and drastic change in social norms and day-to-day lifestyle during this time period. This correlation could possibly be explained by a decrease in transportation emissions and a decrease in burning of fossil fuels. Our null hypothesis is that there will be no change in New York City CO2 levels before or after the pandemic. The two main objectives we had were to collect current data using the egg in Staten Island and to compare this data with the data collected before the pandemic.
Our variables are carbon dioxide levels, and our proposed constants are how often the egg takes data.
In our project scope, we have made the assumptions that the past data sets are accurate and have not been tampered with. Some limitations we have come across are that the project has a limited time frame and data could only be collected for approximately 1 month. And the data collected before the pandemic is global data, whereas the data being collected during the pandemic was measured in New York City. A risk of this experiment is that past data sets could have been tampered with. For our statistical design, we only did one trial, and our sample unit was carbon dioxide. We had 2 sample sites, one in Mauna Loa Observatory in Hawaii, and the other in Staten Island.
The 3 main materials that we used in this project were our air quality eggs, which we used to measure air quality levels and specifically carbon dioxide levels inside or outside of our homes. The air quality egg data portal, which is the site that we use to access our data that we got from the eggs, and the computers, which generally allowed us to bring everything together, create the slideshow, present everything, analyze the data, etc. Our methods consist of comparing 2 sets of data, one of which we collected ourselves through our air quality eggs, and the other coming from a research center in Hawaii. We wanted to use data from before the pandemic to see if there was any real changes in carbon levels throughout the entire event.
These results were taken from the Mauna Loa Observatory in Hawaii, spanning from the years 2000 to 2021, and we can see in the graph a very gradual but consistent increase in CO2 over the years.
This is our data taken from New York, and we can see that the highest CO2 recorded was around 2,135 ppm, while the lowest recorded CO2 we saw was roughly 435 ppm. We can also see a few abnormal spikes in CO2 around March 8th, April 12th, and May 1st, all averaging around 1,000 to 1,500 ppm. Our hypothesis was not supported by our data, and there was no significant change in CO2 levels in correlation to the COVID-19 lockdown. There are a few reasons why our hypothesis was not supported, one of which could have been that there were a few logistical inconsistencies with our project. For example, the 2 data sets that we compared had been taken at vastly different altitudes, locations, and humidity levels, etc. Another idea worth bringing up is that New York City may have been affected harder, so it could have been easier to see the differences in air quality.
And for a conclusion, as shown in the data, there were no significant changes in carbon dioxide levels due to the COVID-19 pandemic, and therefore the hypothesis was not supported. Levels were mostly stable or gradually rising before and throughout the pandemic, despite the drastic differences in both location and elevation. This is our bibliography, and thank you.
All right. We'd like to thank the team who created this video. We also like to thank our post-secondary partners, the Borough of Manhattan Community College, for your support with our after-school program, Harbor Seals. We are also grateful to Dr. Muhammad and Mr. Natan for their support and technical expertise throughout this program. As well, before we'd like to also thank you to our sponsor, Con Edison, and our industry partner, Hudson River Foundation, for believing in our work.
Hello.
Is there a relationship between PM 2.5 air quality and asthma cases in neighborhoods throughout NYC? We are part of the Harbor Seals program from the Urban Assembly New York Harbor School. Our advisors being Mauricio Gonzalez and our group members being Maddie Novak, Marifer Sanchez, Sebastian Coco, and Dainara Sanchez.
As the human population rises in urban environments, the amount of particulate matter, also known as PM, increases in the air. PM are particles found in the air as a result of natural or anthropogenic activity such as a fire or spraying aerosols. PM can be categorized by size, the smallest one being PM2.5, which measures 2.5 micrograms, while the largest, PM10, measures 10 micrograms. PM2.5 has been linked to hospitalization for respiratory and cardiovascular diseases and even leads to death. It has been observed that early exposure of PM2.5 has been associated with severe cases of asthma later on in life.
The finest particulate matter of PM2.5 can penetrate deep into the lungs and even enter the bloodstream. This present study will focus on PM2.5 due to its frequent impact on human health. The air quality egg will be used to collect PM2.5 indoor pollution data in neighborhoods throughout NYC. This meta-analysis will compare air quality data to NYC GovHealth records to examine a potential correlation between indoor PM2.5 pollution and asthma cases in NYC neighborhoods.
Furthermore, leading to our scientific problem being, what is the correlation between indoor PM2.5 pollutants and asthma cases in neighborhoods throughout NYC?
It is hypothesized that asthma rates will increase within each monitored neighborhood as PM2.5 concentration increases. Alternatively, our null hypothesis states that asthma rates will decrease within each monitored neighborhood as PM2.5 concentration increases.
The boroughs in this experiment are Brooklyn, Bronx, Manhattan, Staten Island, and Queens.
Moving forward to our project design, with having our independent variable being particulate matter, while our dependent variable being asthma rates, and furthermore proposed constants being city environment.
Our current limitations are our limited sample size, which lead to a very inaccurate representation of PM2.5 concentrations in different neighborhoods. Our second limitation is our outdated data source surrounding asthma cases in different neighborhoods. Our current assumptions are asthma rates in different neighborhoods where PM high readings are found. Our second assumption is that the data collected from the air quality egg is reliable. Lastly, we are also assuming that asthma data sources are reliable.
Procedures. Set up egg air quality machine. Download data onto sheets. Graph data sets. Create trend line inside the graphs. Use trend line to create an R squared. Find correlations between axes using R squared.
Moving forward to our first figure, which is PM2.5 averages versus neighborhoods. Here, calculations of averages of PM2.5 indoor pollution is collected by our air quality eggs throughout different NYC neighborhoods. The highest recorded data point is Sunset Park with 18.4 micrograms per cubic meter, while the lowest being Jackson Heights with 6.5 micrograms per cubic meter.
Figure 2 shows a relationship between PM2.5 averages and asthma data in children cases. The neighborhood with the highest amount of asthma cases in children is Mauritiana Melrose with 63.7 micrograms per meter squared, while the lowest amount of asthma cases reported was in the West Village with 1.8 micrograms per meter square.
Figure 3 represents the averages of the air quality eggs throughout different NYC neighborhoods compared to the amount of adults that got hospitalized from asthma out of 10,000 in each neighborhood. The neighborhood that has the most adult asthma cases is Morrisania Melrose with 40 cases per 10,000 adults. The neighborhood that has the least asthma cases is West Village with 1.3 adult asthma cases per 10,000.
Finally, in figure 4, we have asthma data for children versus PM2.5 averages. The calculations of the PM2.5 collected again by our egg quality eggs were to the amount of children that got hospitalized from asthma out of 10,000 in each neighborhood.
Figure 5 shows a calculation of the average PM 2.5 collected by the air quality egg throughout different NYC neighborhoods, compared to the amount of adult hospitalization from asthma out of 10,000 people in each neighborhood. It is observed that the R squared is 0.048, showing a minimal correlation between adult asthma cases and PM 2.5 averages. Figure 5 shows an R squared of 0.048 in adult asthma cases, while figure 4 shows an R squared of 0.043 in children asthma cases, showing minimal correlation between the asthma cases in different neighborhoods and indoor PM 2.5 pollution. Mauritiana Melrose has the third lowest PM 2.5 average, while also having by far the most adult and child asthma cases, with over double the amount the highest neighborhood. Figure 1 shows the highest PM 2.5 averages despite having the third least asthma cases in Sunset Park.
Wrapping up our presentation, there were no correlations between asthma cases throughout NYC neighborhoods and PM 2.5 indoor pollution, and so our hypothesis wasn't supported by our data results. Some suggestions for future research is having more data samples and replicates throughout the city, as well as further investigations regarding particulate matter impact on human health, and finally an updated health report on asthma rates, as the most recent one we were able to find was from 2017. Moving forward and finally to our bibliography, accrediting and citing all sources that were used throughout this research project. Thank you so much for listening and have a wonderful day.
All right, so those were our SEALS presentation. Once again, thank you to everybody who worked in SEALS and who participated. We really stuck it through and had a successful SEALS year, all virtual. And just again, I want to say thank you to Con Edison, the Hudson River Foundation, as well as BMCC for helping us and being our sponsors. Just we're so grateful. All right, so now we're going to be moving on to the winning sophomore presentations.
Is rain the cause for fluctuations in the pH at certain parts of the Hudson Raritan Estuary? By Sebastian Coco, advisor Mauricio Gonzalez. What do you call it when it rains ducks and geese? Foul weather. Summary. The data of this experiment was non-conclusive. The available data's limited time span of 2.5 years, small geographical range, and some gaps of months in the graphs made it a challenge to analyze. Background. Rain. Liquid precipitation when water forms around microscopic materials called cloud condensation nuclei. Raindrops fall when clouds get overly saturated. pH. How acidic or basic a water source is. More free hydrogen ions make water acidic. More free hydroxyl ions make water basic. Normal clean rain pH is between 5.0 and 5.5. Acid rain, formed when water condenses around pollutants such as sulfur dioxide. pH of typical acid rain is 4.0. Affects acidity of body of water it falls into. Can be a problem for aquatic ecosystems. Counterintuitively, may eat away at rocks whose alkaline particles wash into waterways, making them more basic. Why is this experiment important for marine science? Large deviations in the average pH can be dangerous towards the physical environment and aquatic life, as they have a limit to how much pH change is sustainable before starting to die. People can also be affected by their fish consumption, drinking, and swimming water with pH extremes. Finding where deviation comes from allows us to start working on creating a solution for possible ecological and humanitarian disasters. Methods and limitations. Compile data and create graphs on pH and precipitation from site. Examine graphs for the data of pH spikes, either acidic or basic. Compare precipitation data by date. This project will examine January 1st, 2019 to April 14th, 2021. Limitations. Some of the graphs do not have data for every month. There is a small geographical range of the piers where data was collected. Pollutants may be flowing into the harbor, which can affect pH. As a whole, the project is not novel, but doing it in this part of the Hudson-Meriden estuary is unique. Studies attempting to do the same thing in the same area were not found. Map location of the piers. This is the northern pier. This is the southern pier. Figure 1. Figure 1 is from Pier 26 during 2019. In all of the figures, pH is orange while rainfall is blue. As previously stated in the limitations, sometimes the data is incomplete, as in this slide where pH is only available for 3 months. Figure 2. Figure 2 is from Pier 84 during 2019. The pH is consistent with the exception of one large increase in pH and another large decrease in pH. Figure 3. Figure 3 is from Pier 26 during 2020, is missing data for a lot of the year. The pH scale on this figure varies from 6.8 to 8.8, as it may be different for the others. Figure 4 is from Pier 84 during 2020, is the first slide where there is a relationship of pH to precipitation. Figure 5 from Pier 26 during 2021 has consistent pH with the exception of one drop. Figure 6 from Pier 84 during 2021 has a pH scale that is so small that noticeable changes are not worth examining. Results. Figure 1 does not show any relationships between rainfall and pH. There is both heavy rainfall and pH change, they are just not at the same time as each other. Figure 2 does not show much in the terms of relationships, but there are 2 exceptions. There is a large amount of rain on March 24th, and at the same time there is a massive dip in the pH to approximately 0, which is considerably more than any other drop on this map. The other change is on January 22nd, where the pH raises from 7 to over 15. Whether these 2 changes were from the rain, dumped chemicals into the harbor, or malfunction in the machines doing the testing is unknown. Figure 3 does not show any major relationships between rainfall and pH. Figure 4 is the first figure seen so far with a decent amount of evidence for a relationship. On the dates September 4th, September 10th, and December 16th, there are changes in the pH as well as in the corresponding rainfall. Figure 5 does not give any relationship between rain and pH. There is one large drop in pH during March 19th, but it does not coincide with any rainfall. Figure 6 is generally does not have much in the way of relationships. Conclusion. Generally, the data shows that the rainfall does make very small changes in the pH, but these changes tend to be small enough that they would not affect most animals. When it comes to large changes that could have more an effect, there is not much data to support it. Generally, the flaws of the experiment, such as a lack of complete datasets for full years, along with only looking at a time span of a little under 2.5 years, makes it hard to pull a conclusive answer. Future steps for other scientists. There are many ways someone could expand on this experiment, with one being the increasing of the range of the area being studied. Adding on more of the New York Harbor or going further up the Hudson River could greatly increase the value of the study. Another way to expand on it would be to move the entire location to somewhere else, but still use the same type of methods. This may allow for more data to be discovered. Overall, the main next step would be taking the methods of the experiment and testing on other things that could be possibly affecting the pH besides rain, for example, outside pollution. Thank you for watching this presentation.
All right, okay, so that was our winning sophomore presentations. And now we're moving on to our winning junior presentations. And I also would just like to thank the sophomores. I would also just like to thank the sophomores as well for persevering through this year and getting in all their presentations. So thank you guys. And if you have any questions about any of the presentations, leave them in the chat.
You would think your baby is safe from all outside harm while in utero. But imagine this. What if there were tiny pieces of plastic floating around in the womb next to your baby? Unfortunately, there was no need to imagine for 6 mothers in a study conducted by Antonio Ragusa at the San Giovanni Calabata Fatabenatelli Hospital in Rome. The study observed microplastics of 10 microns in the placentas of newborn babies. If you needed another reason to believe that microplastics and plastic pollution in general are a huge problem we must deal with, I would look no further. But what if I told you there was a solution? My name is Marifer Sanchez, and this is polyethylene degradation by bacterial strains. In 2015 alone, the global plastic production rate exceeded 322 million tons. That is more than it has ever been in the 25 years prior. This is causing major alarm for terrestrial and marine resources. An increase in global plastic production rates leads to excessive amounts of plastic pollution worldwide. Of that pollution, polyethylene, also known as PE, is the most prominently observed plastic pollutant. PE has been found anywhere from agricultural fields, intestines of cows, and even secluded tundra biomes. The frequent appearance of this microplastic is slowly proving to become a threat to not only our food sources and health, but also our aquatic and terrestrial environments. To help find a solution, the present meta-analysis will compare different PE-degrading bacterial strains to identify the most successful in degrading PE. Successful degradation will be determined by the achieved weight loss and topographical modifications done onto the PE samples by each bacterial strain. This data will be collected from a variety of studies. The question this study will answer is which of 37 bacterial strains will be the most successful in degrading polyethylene, also known as PE. There are 3 major objectives in this study as of now. Number 1 being to search for peer-reviewed journals, also known as PRJs, on bacterial strains with PE degrading capabilities through the literature review process. Objective 2 is to categorize and identify each bacterial strain's effect on PE. In other words, the bacterial strains with the most effect on the weight loss of PE will be grouped together, while the bacterial strains with the most effect on the topography will be grouped separately. Lastly, Objective 3 is to compare each bacterial strain's effect and identify which one is the most effective in degrading PE according to this study's definition of biodegradation. Now let's take a look at all the methods that will make this meta-analysis possible. To resolve the first objective, the first procedure of this meta-analysis is the literature and data collection. Step 1 is to use a reputable publication site such as Elsevier or ResearchGate to obtain literature and data on different PE degrading bacterial strains. Step 2 is to create a space of storage to sort the said literature and data by bacterial strains. In other words, all of the associated Enterobacter aspergilli data and literature will be grouped together, while all of the penicillin data and literature will be grouped separately. To resolve Objective 2, the second procedure is the organization of data. Step 1 is to create a spreadsheet tracking PE dimensions such as initial PE sample length and width, initial PE sample thickness, and the same dimensions for the modified PE samples. Modified refers to the modifications done onto the sample after being degraded by the bacterial strains. Step 2 is to extract data from the previously organized PRJs and organize it into the spreadsheet for further analysis. The project scope focuses on the limitations and assumptions of this study. Currently, there are 3 limitations in this study. One being that each level of error within different experiments may interfere with the results in this study. Another limitation is the lack of controls. The last limitation is that some variables may not be comparable to each other. 2 assumptions made in this study are 1, that the previous mentions of the bacterial strain degradation are accurate, and 2, that the PE tested in each experiment is similar enough to not alter the results in this study. Meta-analyses require much time behind a computer, which doesn't really pose any immediate risks, but to ensure maximum security and protection of ethics, researchers must trust that the studies they find follow a reputable code of ethics. In addition, caring for one's mental health is very important as workplace stress leads to clutter, emotional exhaustion, and decision-making delay. Finally, researchers must make sure that their surroundings are clear and safe. My name is Marifer Sanchez, and this has been Polyethylene Biodegradation by Bacterial Strains. Thank you.
Okay, I'd like to clarify that this is a proposal. All the junior projects are proposals, and so Marifer will be working on this project throughout the next year.
All right, so thank you guys to our juniors who were presented. And now, last but not least, it'll be our winning senior presentation.
The code for this project is Indy06. Hello, I hope you're having a good day today. My name is Max Feldman, and today I'll be presenting on the possible discovery of a new species of Fundulus in the New York Harbor. I conducted this project with my mentor, Louise Bott, at the American Museum of Natural History, and I'm a student at the New York Harbor School in the Marine Biology Research Program, and I graduate in 2021. In the summer of 2019, a project was conducted to try and answer whether or not Superstorm Sandy had an effect on the population genetics of Fundulus heteroclitus. In this project, the non-coding mitochondrial control region was analyzed since it gives specific information on maternal and population genetics. This was going to be used to try and see if Superstorm Sandy had any effect on the population genetics of these fish. The study was done on Fundulus heteroclitus since it's an abundant tidal fish located along the East Coast and in the Hudson Estuarine. And as an abundant tidal fish and generalist predator, Fundulus heteroclitus is an important organism in its ecosystem and provides information on overall ecosystem succession. For example, it's prey for white perch and blue crab, and it's a predator for grass shrimp and snails. So it's in the middle of the food chain in its habitat. Fish were collected at 4 sites across the New York Harbor, one being Inwood Hill Park, another Jamaica Bay, Jones Beach, and Pelham Bay Park, and then after were brought back to the lab for DNA extraction using the QIAGEN blood and tissue DNA extraction kit. After DNA extraction, the samples were put through the process of PCR to copy the DNA many times and then amplify it to select a specific region of DNA, in this case, the mitochondrial non-coding control region. And then the samples were sent to a company called GeneWiz for them to be sequenced. Once these sequences were returned, they were uploaded to a software called DNA Subway, which is connected to GenBank to compare our sequences to other Fundulus sequences on GenBank. So each sequence that had base pair mismatches showed around 4 to 10 base pair mismatches and had blast hits, which is when the sequences are being aligned with GenBank, and they matched up with 2 Fundulus species that are native to Midwest freshwater habitats, one being Fundulus julissea and the other being Fundulus sciaticus. So this data, while it didn't show any evidence of Sandy having an effect on the population genetics, which means the research question is inconclusive, is important since it shows that there might be a new species of Fundulus or freshwater Midwestern species of Fundulus in the New York Harbor, which is extremely intriguing. And it might be possible that these base pair mismatches were due to genetic mutations or environmental differences. So the plan before COVID-19 was to go back to the already extracted DNA and then resend them, re-PCR and with a different primer, and then send them out for sequencing to get a new sequence of DNA. And in this project, a different sequence would be taken to try and identify these unidentified samples. So since COVID continued throughout the summer, research was entirely online and no lab work can be done. And so during this summertime, a comparative study was done where the samples from this project and the samples from a different project, which was very similar, were compared. And in this man's project, Andrew Whitehead, he found that there were around 6 base pair mismatches per sample of his, and he said that that was due to genetic differences, mutations, maternal differences, or environmental differences, but not a new species or anything like that. So this year, since COVID restrictions were not as bad, the project was able to be conducted, and it was conducted through the Urban Barcode Project. So in this project, the goal was to see whether or not there was enough base pair mismatches in the barcoding COI region to describe a new species of Fundulus, since the barcoding COI region is used to identify fish species. And it is hypothesized that there will be enough base pair mismatches in these sequences to identify a new species of Fundulus. So during this project, already extracted DNA from the American Museum of Natural History was taken to Urban Barcode Project labs for PCR protocols using the fish vertebrae primer, and gels were performed on the already extracted DNA from the previous project by Louise Bott, since I was still not allowed to go into the lab, but she was able to do the lab work for me. So after the lab work was completed, the samples were then sent out to GeneWiz for sequencing again, and this time they were going to be sequencing the barcoding COI region for us, which is a much larger region. And once the sequences were returned, they were analyzed and graphed on DNA Subway again. So for the results, there were 28 total successful sequences, and 11 of these sequences, which were the samples that had the base pair mismatches in the initial project, all turned out to be another type of Fundulus species called Fundulus majalis, which is a very similar striped killifish species to Fundulus heteroclitus. And the remaining 17 samples that all had 0 to 4 base pair mismatches compared to Fundulus heteroclitus sequences from GenBank. And in this phylogenetic tree, all the samples were grouped based on their blast hits, which is when the sequences were aligned with GenBank sequences to be compared. As you can see in the top, these are 11 of the sequences being grouped with the Fundulus majalis GenBank sequences. And then down here in this big group, there are the remaining 17 sequences being grouped with previous GenBank Fundulus heteroclitus sequences. So based on the data, all the samples have either been identified as Fundulus majalis or Fundulus heteroclitus. So based on this, the initial hypothesis is rejected because a new species of Fundulus was not described. However, this data is still valuable since it may be some of the first mitochondrial non-coding control region sequences of Fundulus majalis published, which is why there may have been mismatches in the original project. And also, the 1 to 4 base pair mismatches that occurred in some of the Fundulus heteroclitus samples is not significant at all, and they can still be identified as Fundulus heteroclitus since this region is much larger than the previous region sequenced. In addition, back here in this phylogenetic tree, these outlier groups, the fact that none of our samples are had blast hits with the outlier groups further prove that they're either Fundulus heteroclitus or Fundulus majalis. Thank you for listening to my project, and I hope you have a wonderful day.
All right, and that was our very last senior presentation. I think now Mauricio will take it away and talk about our award ceremony.
Okay, congratulations to all of our research scholars. We are 10 minutes ahead of schedule, so I'm going to ask you all to take a short break at this point, and we will reconvene at 3:10, where we will announce the remaining winning projects of our program-wide symposium. At 3:10, please come back. Okay, for those of you that are just joining us, we are taking a break. We will be back at 3:10 for the award ceremony. That's in 5 minutes. Okay, we'll begin in about a minute. Okay, Carla, if I can have that next slide, please. So welcome to 10 years of marine science at the New York Harbor School. This is the moment you've all been waiting for, where we get to announce the winners of the symposium. I'd like to take this time to thank all the judges that dedicated a week of their time to review projects. And all of this was virtual. And for the patience that you all demonstrated as we figured out how to deploy this big undertaking, and soon enough we have the results, and soon enough your thoughtful comments will be shared with our scholars so that they can improve and get to that point where many of our scholars now are in their PhDs or graduating from undergrad degrees in biology and so forth. But first, I'm going to bore you for about 6 or 7 minutes to highlight some lessons learned and some of the accomplishments that the MBRP has had over the years. Research is different. It's a game changer at the high school level. When I was growing up, knowledge and content was obtained by going to institutions and receiving lectures or textbooks from adults. They acted as knowledge gatekeepers. Today, though, students have all the knowledge of the world at their fingertips. This is important because the questions are changing. The questions are no longer, what can I learn from you necessarily, but what can we accomplish with the knowledge that we have? Even further, questioning is becoming, how can we gather our own knowledge and use it to make a difference in an ever-changing world or economy? Schools, unfortunately, are still stuck in yesteryear's gatekeeping of the what. But we need to build schools that can deliver the how and have the vision for the why. This is where project-based learning, CTE 2.0, portfolio schools, and grading for equity come in, or as I prefer to call it, phenomenon-based learning. This has traditionally been the role of higher education, but it's increasingly becoming the role of high schools. Students are becoming more conscious of the situation and are becoming more eager to solve problems around the world and in their communities, rather than sit and pretend that a teacher is the gateway to knowledge. Changing the world is taking center stage. But the internet is filled with erroneous and misleading information too. So how does a young person learn how to differentiate these? The good information from the bad information? Well, that's where research and experienced mentors come in. That's the new role of our teachers, to share that experience so that they know how to do research. Again, this is what I call education 2.0 or CTE 2.0 or phenomenon-based learning. However, there are big obstacles to research and phenomenon-based learning. The DOE and schools are still trying to fix a lot of yesterday's schools. Many folks don't believe in our kids and have grading biases that keep our most vulnerable students behind. Our economies need to adapt to tomorrow's jobs, like green or blue jobs. We need restoration technicians, green and blue infrastructure technicians, harbor cybersecurity technicians, harbor cyber modeling for climate change and its impacts, transportation management. We even need urban vertical agriculture centers for local food production, green maritime transportation, and on and on. There's so many issues and so many opportunities to tackle in today's world. And in the most powerful city in our universe, as I like to sometimes tell my students, as far as we know, we need leaders who can implement a bold, new, blue, and green vision. Our scholars depend on it, and our city depends on it. It's been 10 years at the New York Harbor School, 10 years running symposia, and it's actually my 17th career symposia. Carlo, if I can have the next slide. 18 years ago, I was put in charge of managing a new greenhouse in a courtyard at a great school in Harlem called the Frederick Douglass Academy. Our first experiment consisted of testing the effects of substrates on seed germination. And little did the school know, or anyone else, that we would soon be building one of the first urban vertical agricultural programs in the city. Next slide, please. Our first projects may have been simple, but they helped lay the foundation for a burgeoning high school research program. Next slide. My middle school and after school students began designing and building recirculating hydroponics and aquaponics systems, the first in the New York City DOE. My high school students designed and built an elevated garden in the courtyard, complete with a fully automatic irrigation system. They were then called upon by various neighborhood organizations to build systems for them. Next slide. This is a picture of our first symposium team back in 2014. Next slide. And since then, we've grown and started making a splash in the world of marine science. Over 28 scholars have competed in the regional symposium. Assuming that there's about maybe 400,000 high school students and 500 compete yearly, that's only a 0.125% chance of a student competing and having access to these symposia. Could you go back, Carlo? Thank you. "126 scholars have come through the program. Andrew Sommer's class being, I believe, the third class. And 92 of those have received their CTE stamp, which is a school high. Over 550 college credits have been awarded, which is also a school high. And over 90 internships have been worked, or 90 students have been in internships, which is another school high. That's the kind of work that you can't make up, kind of like how this next picture, if you can share that picture with everyone, Carlo, that's how these hats get worn out. And you can't make this up, right? This has been a tremendous amount of work, and I would like to share that all of this could not have been possible without all of your collaboration and your participation and your hard work, and especially those that have not given up, those that continue to struggle and move forward and help the team and step up and take leadership and communicate effectively and believe that they can do it and are ready for a challenge, because I would have to say that this program is incredibly challenging, and I am a person who challenges my students as best as I possibly can. So next, Carla, I want to take you down memory lane here and talk about previous themes. I thought this might be interesting to share with you all. So this first poster theme was created by Jeleni Abreu, and the second one was created by myself. Next slide, please. The third was designed by Pablo Jimenez, and the fourth by my wife, Anita Morawski. And by the way, my wife has stepped up numerous times to support the development and success of this program. So thank you, Amor. And by the way, it was her birthday 2 days ago, so join me in wishing her many, many more, filled with lots of health. Stolat. Next slide, please. The fifth was designed by GIS scholar Julia Montilla, and the sixth by myself, based on a photo shoot taken by the artist Van Wong of my students in action. The seventh was designed by alumnus Matthew Chu, and the eighth by our Trident Award winner and alumnus Lauren Salatan.
Never give you any answers because they always, what they say is, well, we hope.
The ninth was designed by myself during the pandemic last year based on a drawing by the Hill Fox Art Series and various depictions of the COVID virus, HAL from 2001: A Space Odyssey, and others. This year's theme was designed in whole by our class of 2021 scholar Emily Shee. Okay, and now really for the moment that you've all been waiting for.
All right, okay. So I would just like to announce the categories, which is going to be the 10th grade categories are aquatic ecosystem. The 11th grade categories are AMB, oceanography, and INDY, independent research projects, are 2 tracks. And then for our 12th grade categories, we're going to have GIS, geographic information systems, and INDY, independent research projects.
So for aquatic ecology, our 3rd place winner is James Karsh. Please hold your applause till the end. 2nd place, Julia Purazella. And 1st place, Sebastian Coco. For our advanced marine biology and oceanography 11th grade scholars, 3rd place, Bryant Soriano. 2nd place, Faith Yee. And 1st place, Chelsea Clark. For our independent research scholars in the 11th grade proposal category, we have Maddie Novat in 3rd place, 2nd place, Heavenly Davis, and 1st place, Marifer Sanchez. For our 12th grade GIS scholars in 3rd place, we have Gabriel Castro, Pedro Vieira, and in 1st place, Luke Sampton. For our independent research 12th grade scholars in 3rd place, we have Prophet Davison. 2nd place, Kate Mumford, and in 1st place, Max Feldman. And here are the names. I'll give you all a minute, and if you would like to unmute and give our scholars, after a huge year of COVID and sticking with science, you may unmute yourself at this point and give them a round of applause.
Congratulations, you did it!
Okay, and this would not have been possible without the support through the years of many of our post-secondary and industry partners. So the Marine Biology Research Program would not be successful if not for their generosity and dedication that they show year after year. Putting their confidence and belief in our students and our staff is a noble act that can only be explained by deep commitment to the stewardship of our natural world and the wish for the success of our community and city. They know the future is in the hands of our young scholars, and it is with that dedication that they offer them opportunity after opportunity. Whether we embark in new partnerships with BMCC to develop a CTE 2.0 career pipeline, funding and helping to administer our ambitious SEALS program, finding guest speakers to come talk to our budding young scientists, or doing all the behind the scenes work, keeping the lab running and our organisms alive during the height of the pandemic, even until today. Thank you for all of your support. We are honored to have you all as our NBRP family and PAC members, and hope to continue to partner with you as we continue to prepare our youth to explore new and unknown frontiers. We have Mr. Benito Nunez, our New York Harbor School custodian, who has been our lab technician for the last year and a half. Dr. Mohammed Azhar from BMCC, partnering with our after-school program. Dr. Hao Tang from BMCC, and Dr. Chin Song Wei from BMCC with our GIS program that our students recently completed courses with at BMCC. Dr. Matthew Haken from the Billy Noyster Project and as the funding officer. Mr. Michael Porto from Con Edison and Mr. Joshua Crespo from Con Edison, who believed in our students and in the program over the last several years, without whose support I cannot stress we would have had a lot of difficulty being as successful as we are without their belief in us and without the support that Con Edison has given us. And last but not least, Ms. Kelly Knight, one of our graduating MCs, Prophet Davison, her mom, and for her support throughout the years, moral support, and for giving us a wonderful student that has certainly helped this program flourish. Thank you to all of our special honorees. And if any of you would like to unmute at this point and share a couple of words with us, you may do so.
I would just like to say that, thank you to everyone who's on the special honorees list, and as well as all the people who deserve to be honored, who are behind the scenes, whether that means students, parents, or other. We're just really thankful for you guys to help and just believe in this program so much, and we wouldn't be able to make such a strong program happen without you guys.
Okay, I'd also like to emphasize, yeah, I think I hear someone who may want to share a couple words.
Yeah, Gabriel here. I just want to be thankful also because most of my work is lab tech and I wasn't able to go in for most of the year. So I particularly want to thank all the people who stayed behind to do all that work, because that is truly invaluable, because all those organisms need constant maintenance. And I should know, since I had to work with them from day until early in the morning until late at night. So I want to say thank you for all the work you've done for the past year.
Yeah, and I may want to add that it was very difficult for our students when they came back to the school to see the lab and to not want to be a part of it and to help it come back to, to improve upon it. And unfortunately, it's really difficult to open up a lab space and allow students to engage without the appropriately trained personnel. So I understand how difficult it was and it is. And so what I'm hoping is that next week, when I do go to the lab, that you will all accompany me as we start chipping away little by little to improve the lab again, and give Benny some support, who's really done outstanding work there keeping the organisms alive, and also all of our students, to celebrate all of our students, because as I tell all my students every year, any one accomplishment is done in thanks with the help and support of everyone else. And so if we have these winners, it doesn't necessarily mean that they did everything alone. It was real because of the work of all of the other students. And so if the student body it doesn't raise itself and put itself together, we can't have these successes, big and small. So thank you to all the students. And I see a couple of hands up. So Kelly, would you like to add some words here?
Yeah, thank you. I just wanted to say thank you to you and all the students for recognizing my small contribution. And also thank you for keeping science interesting. And although the lab wasn't open, I was happy to bring the lab into my home this year. And it was a real education for me also. So I thank you for keeping science interesting, even for an old lady like me, learning all of those things over again, some things, of course, that I didn't know at all. And I look forward to seeing the program continue to grow, although I am, a parent of a graduating senior. Maybe at some point in the future, if my help is ever needed, I'll reach out to Nan.
Thank you. And Kelly, I didn't know that you were another one of my students.
Totally.
It's been a pleasure.
Well, yeah, 4 weeks with 4 tanks of oysters in my dining room. I'm definitely a student now. Not graduating though. I'm sure I didn't earn enough credits for my
Wonderful.
small education. But yeah, it was really nice to see up close and personal how the marine bio program works and how important the hands-on part of the learning is. So thank you, and thanks to the students. It's a wonderful symposium again.
Thank you very much, Kelly. And we actually have our next guest speaker that just signed on. So Dayanara, would you please take the stage?
Hi everyone, I'm Dayanara Sanchez. Can you hear me?
Yes, we can.
Okay, great. So we are very pleased to have here with us today as our honoree, the Manhattan Borough President, Gail Brewer, who we laud for her longstanding support of young people across New York City. Gail is the 27th Borough President of Manhattan with a huge role in deciding land use issues in the borough, appointing members to the 12 community boards, and with ability to introduce legislation in the City Council. She previously served on the council for 12 years, including as chair of the Technology Committee. Gail earned an MPA from Harvard Kennedy School of Government and did her undergraduate work at Columbia University and Bennington College. A ceaseless champion for all students in our city, she has made her particular focus for equitable access to technology and expanded mastery in STEM, science, technology, engineering, and math for young New Yorkers like us. President Brewer has, in her 8 years in office, directed hundreds of millions in capital funding to projects ranging from new school science lab and internet infrastructure to improving cultural institutions. Her dedication to greater STEM opportunity is especially meaningful to Harbor School, as the majority of our youth represent diverse ethnicities and more than 1/3 are young women. In fact, Gail has been a steadfast supporter to Harbor for all of its 11 years here on Governor's Island. She has presided over graduation and recently supported our new expansion and building requests on Governor's Island. She has been quoted as saying, "Research and common sense both tells us that experiential education opportunities produce deeper learning and more meaningful experiences for students. Harbor is all about hands-on project-based learning, of course. We in the Marine Biology Research Program are simply delighted to have her with us today to celebrate the culmination of years' work in marine science in the context of one of the greatest scientific experiments in resilience, which investigates how living things weather adversity. And as she completes her term as the best borough president ever, we hope that she will return to again serve in the city we all love, and what will undoubtedly be some rough waves ahead. And now I will be passing the mic over to Madam Gail Brewer.
Borough President, there is a mic symbol. It is at the bottom of the screen that you will need to hit to unmute yourself on Google Meet. Okay, we may be having technical difficulties. And I can continue with this next part of the presentation. And if you can unmute yourself, there's also a chat. So if you would like to maybe let me know through the chat if you can't find the microphone function, and I can continue with the presentation here. Okay,
Mauricio, is it possible that you can manually unmute her?
That's a good question. I'll try. Let's see. No, I don't think I can. All I can do is remove or mute, but I can't unmute. Okay, so So thanks, Gabe, for that suggestion. In the interest of time, if you can unmute Madam Borough President, please let me know. And Nan, if you can, if behind the scenes there, if you can communicate and maybe let me know whether I see two accounts for Gail Brewer that just showed up. So maybe it's a computer issue.
I just contacted Gabrielle, her schedule, to ask her to unmute. So let's see if that message got communicated.
Okay. In the meantime, actually, why don't we hear from? I know that there were that Dr. Nolan and that Anita Morowski had their hands up. So do you folks want to express any ideas right now? And I apologize that I didn't move to you, but I was trying to hurry things along here. So if you'd like to chime in at this point, maybe this is a good time for you to do so.
I'd like to say something, Mauricio. First of all, thanks for putting together a wonderful program, and thank you, all you students. Great. I mean, I love the data-driven projects that you did. I forgot to mention at the end of my talk that we have a program at St. Francis College that runs from July 6th to it's a 2-week program. It's free and it's virtual this year. It's our second year doing it virtual, and it's called the Summer Science Academy, and it runs like from 9 to 2, I think. So if you want to apply to it, you can send me an email, which is KNOLAN, pretty easy, K-Nolan, at sfc for St. Francis College.edu. And your Professor Gonzalez has the email anyways, so.
That's right. That's right. And
They're
I will
free program. Yeah.
I'll forward that information to all the students. And okay. And do we have any other messages? Any other folks who may want to chime in? Okay, so I'm gonna move ahead with the program. If anyone thinks that's a bad idea, let me know. But I think that we should move ahead. These technical glitches can take a long time, and everyone's pressed for time here. So let me first start announcing and talking a little bit about the MBRP Trident Award, the Marine Biology Research Program Trident Award of Excellence. This award goes to a scholar that has 1, earned all possible college credits that were offered to them in the program, 2, competed in the regional science fair, NICEF, Terra, or completed GIS certification, and 3, has demonstrated outstanding leadership for 2 years in the SEALS Civic Science Monitoring Program. Next slide, please. Past MBRP Trident awardees have been Nicole Martinez, Cezanne Bees, Melanie Smith, Marcus Charles, Anila Allen Sutherland, Sid Bloomfield, Lauren Salatan, and last year, Jonah Flor Holman Bauman. Next slide, please. And this year's Trident Award winners are Gabriel Castro and Kate Mumford. Congratulations to our 2021 Trident Award winners.
Woo! Yeah, you're
there. Okay. Next slide, please. Okay, and our closing remarks. Thanks to our New York Harbor School Parent Teacher Association, New York Harbor School administrators and staff. Next. And our MCs, if you want to step in at this point.
We just want to say thank you to all our industry and post-secondary partners, such as the BMC Foundation, Earth Matter, Con Edison, the Nature Conservatory, Digital Quest, among many others, and NYU as well.
Okay, and I am, I have been blown away. I think this is the best organization, the best teamwork and leadership that I've seen in a very long time in these incredibly difficult times. So class of 2021, you will be missed. I'd like to read off the names, and so that all of you can see who they are. We have Gabriel Castro, Carla Cortez Reyes, Prophet Davison, Max Feldman, Oliver Lieber. Emily Lysakova, Sarah Mazzoli, Kate Mumford, Daniel Picarello, Dakota Rogers, Luke Sampton, Dayanara Sanchez, Emily Shi, and Pedro Vieira Costa. Jasmine Mendoza, Ayana Avery, Justin Jaquez, Penelope Fernandez, and Gideon Brown. Thank you to our whole Harbor and MBRP community. The ceremony and accomplishments of the MBRP would not have been possible without your dedication and hard work. It literally takes a whole community of MBRP friends, family, professionals to make this possible. It takes each individual pulling their own weight and keeping their plan together. And each successive class has benefited from the hard work and dedication of our alumni. Those coming after you will also be benefiting from your fruits. From earning 21 college credits, working on independent research projects with professional mentors, working with cutting-edge GIS software, operating world-class recirculating aquaculture systems, internships with our industry partners, conference presentations with our post-secondary partners, meeting in virtual NBRP labs to make virtual learning easier, to competing in the city with the city's best science schools and students. No one here has accomplished any of it on their own. So thank you to all of the students that have not, even those that have not been mentioned here today and are working behind the scenes, like our 11th graders and our 10th graders and our 9th graders, as we speak, alongside those that have been mentioned today in this presentation.
I'm here. It's Gail Brewer finally, but I give up on the video.
Okay, Carla, can you go back?
Grew up like 5.
Carla, can you go back, please? Can you go back to just one slide? Thank you. Okay, Madame Borough President, thank you. You may speak. You have the floor.
Thank you very much. I apologize. I don't know why my cell phone doesn't give me the ability to both video and speak, but I want to thank Dayanara Sanchez, who is not only an amazing presenter, but also a great producer. And I also want to say, as you know, how great the Harbor School is, not just recently when you managed to pull off a building and a pool, but also as a long time high school that is unique and has an amazing student body and principal and staff. So congratulations. So this issue of marine biology, obviously the pool will help, although you're going to have a long way to actually get there. I do want to say that when I was in the city council thinking about STEM, I did start with the American Museum of Natural History, the Urban Advantage. And what that does is in every borough, there's a museum of scientific interest, obviously the botanicals and the American Museum and the Museum of Science in Queens. And each one of them is trying to work with the middle school students on a project that then ends up at the American Museum of like 3,000 students and their projects. Harbor School is just doing it again. And these are the kinds of projects that make a difference in people's lives. I know some of your graduates quite well and how they're getting their marine biology, not just interest satisfied, but also getting captain's licenses, some of whom are young women, first time ever. It's a pretty amazing history of success that you are pulling off and being so unique. But anyway, the fact that there's a science fair, that you've got GIS certification, that you figured out how to do civic engagement as part of science, and that Kate Mumford and Gabrielle Castro have also gotten the award. It's like something miraculous that you're pulling off at the Harbor School. I think the students know it, but maybe not all of them. Certainly the parents do. They're so enthusiastic about the school, and God knows the teachers are awesome. So, you've been called the jewel in the trident as a program, but I have to say that's what you could also call your school. And I know that as we're trying to figure out about post-pandemic, we're trying to figure out about science and housing and healthcare and everything. What is it going to look like? I think if you as an institution were in charge, you would do the right thing. So I'm just here to say that there's nothing better than the civic scientists that you are breeding. Obviously the oysters are part of it, but there are so many other ways. And there's a lot of discussion now about climate change. When George Floyd was killed, we talked about a moment or a movement. And certainly, there's a lot of racial justice discussion, but climate change is part of that. They all go together. And, people are trying to figure out, Governor's Island. I'm not crazy about the new proposal, but you are a good part of it. I don't like the tall building. Everybody knows that. But that climate suggestion that Governor's Island is trying to pull off, you're already doing. So it will be interesting for your future classes to see how the climate change academics taking place on the island also can fit into what you're doing. Because everybody talks about climate change and what we need to do, but it doesn't actually always happen. What about these green jobs? What about, are we really going to make all of our buildings energy efficient? Are we really cleaning up the water? What does that mean for the, all the biology issues that you know better than I, but also what does it mean for resiliency? What does resiliency actually entail? Every time we turn around, it costs $1 billion here and $1 billion there. And we don't know if we're even doing it the right way. We bring over people from Holland to try to advise us because the Dutch seem to keep the water out. But all of this is, obviously you're interested in bringing the harbor back and people can realize that they actually live on an island. I can't tell you how many people, they don't know they live on an island. And it's not funny. People actually say, I say, do you know that there are islands in Manhattan and New York City and that, and they say, yes, Rikers. That's sometimes what people say. That's really scary. So we have to tell people, and one way to do it so they know they live on an island is to have them get to the waterfront and enjoy it. But you can't do that unless your students have figured out what to do. So you're pretty unique. You're pretty fabulous. And I just want to say thank you for being so fabulous. And thank you for actually doing what everybody else is talking about, what should get done. So, and you certainly have phenomenal students and staff. Congratulations to everybody. And I look forward to hearing more specifics about this amazing program, the science and the GIS. I do love GIS. That's my favorite thing to do, map. So congratulations, and thank you for including me, and I'm sorry about my stupid cell phone.
Madam Borough President, I am humbled that you have taken the time of your busy schedule to address us, and I believe that you've said how important our work is, and I want all the students to know that if you can have the time, if you can have 5 minutes of this busy person's schedule in their lives, that is a testament to the quality of the work and dedication that you have all brought to this program. And for those of you who sometimes feel like giving up because it's tough, we all are here to support you. All you need to do is unmute yourself, show up, and say, how do we do this together? That's all you need to do, because we're on your side and we're here to support you. And the whole purpose of this whole thing is to make you and New York City a better place. That's the only reason we're here doing all of this, is to bring back New York City and our waterfront and our marine life and all of our citizens having access to it democratically and without any exclusion. That's why we're all here. And so all you got to do, if you're feeling a little scared to ask a question, to ask for help, just don't be afraid to unmute yourself and do so. Thank you, Madame Borough President, and thank you, Dainara Sanchez, for putting all of the, for inviting our borough president, and for that little dream you had of, hey, could we bring her along and address us? And you did it. You did it, Dee. You got it done. And thank you to all the others. Thank you to Nan and all the parents and our partners. And I think my wife wants to say something.
I do, and I'm coming to this computer because my microphone wasn't working. So I just wanted to, in response to the students who were saying how difficult it was to not be in the lab during this time that Mauricio was teaching remotely, I just wanted to personally thank all the students for their patience, and the staff and administration and parents and partners in industry and universities for your patience with Mauricio teaching remotely and not being able to access the lab. As you probably know, he's teaching remotely because of me being immunocompromised due to a transplant. And so from the bottom of my heart, thank you all for your patience as I recover, and for letting him teach from home so that we can both stay safe. Thank you.
And she is stronger than ever. I can't believe she's stronger than me now. I have to be careful. So, and so, yeah, your patience has allowed me to be able to do my thing so that she can be better. She can recover from her transplant. So thank you, everybody. And, life is good.
Yeah.
All right. I don't know where we are in the program at this point. So okay. All right. And I don't want to turn things too sad, but if we can please have the next slide. And we do have a dedication of two people that we love that have passed. And Prophet, if you can please, and Carla, take it from here.
I grew up like 5 minutes from the beach, and really my dad used to take me snorkeling every week. Even my high school project was about the breakwaters of Tel Aviv, where I live. I've seen beautiful, beautiful reefs, but I've also seen a lot of devastation. I think that with that notion, knowing that climate change is not going to go anywhere, we're going to have more rising seas and extreme storminess, which means that we're going to have a lot more concrete structures.
And that's where we come in. We are enhancing the growth of oysters and tube worms and coralline algae, and like a crust of marine life. So think about a seawall that has a crust on it. When the waves and the sediments try to erode the infrastructure, it's protected.
Another prize that goes to winner in the category Forward Actor Female, Dr. Shimrit Purple-Finker.
We envision that next generation of coastal infrastructure will be nature inclusive, and that when we're protecting our coastlines, it won't be on account of natural ecosystems like coral reefs or mangrove beds. We can build and protect our shorelines without destroying nature. We can think about the ecosystem and about the social needs of better ports and stronger infrastructures.
We'd like to dedicate this symposium to Bev and Shimrit Perkle Finkel for all they did for our school as well as the world. And of course, with the difficult year we've had, to anyone who has lost someone, this year our hearts are with you. We would like to have a minute of silence to acknowledge who have passed and their loved ones struggling through the difficult times. Okay, once again, thank you guys. If you are still here, we really appreciate you. Also, I saw a raised hand. So if there are any questions, I think we have a little bit of extra time, so we will be able to take them. I just wanted to say we would just like to thank everyone that helped in some way, especially during a pandemic. And I would like to say thank you to the parents, any of the administrators. We really appreciate you, and this wouldn't have been possible without you guys. And thank you to the students this year too, because we know it's been a really hard time, especially with everything being virtual and constantly going on Zoom, the waking up early, staying up late on meetings. We really appreciate it all. And of course, to the class of 2021, I hope we all see our greatest college experiences and continue to carry Harbor in our hearts as we move on. Now, I think we have a little bit of time for questions, I believe. I saw somebody raise their hand, or a few questions in the chat.
Okay, I've included a sign-in form for those of you who have not signed in. If you can please sign in, and any comments, you can put it in that form. And if you already submitted the sign-in form, you can go back in your email and add any final comments to it. And Nan, I think you have your hand up there. Unmute yourself. I'm sorry, unmute yourself. You muted yourself again there, Nan.
Sorry, I didn't mean to put my hand up. I was looking for the chat, but
Oh, okay.
thank you anyway.
And that concludes our 10th annual second virtual Marine Science Symposium brought to you by the students from the New York Harbor School's Marine Biology Research Program. Thank you to all our guests, and we will see you next year. And students, we'll see you next week to say our final goodbyes on Tuesday. And for seniors, tomorrow you are taking your exam, your final exam. So please show up at the school promptly. If you don't know what I'm talking about, read your emails. And if I am forgetting anything and anyone wants to chime in terms of next steps, any important next steps. Oh, SEALs. Tomorrow is also the last day for you to submit your payroll hours, your internship hours. So please go ahead and put in all of your hours today, right after this, and put that in the payroll in the CTS system. Tomorrow will be your last day. It's better if you do that today. I'd like to thank Mike Cohen in the background, who was on standby all afternoon to rescue us from any kinds of issues we may have come across. But I'd like to thank Carla, our scholar, who spent a lot of time in the background with our slideshow, our MCs, all of our committees. We had several committees. I'd like to thank our communications committee for communicating with all the various guests that we had, our booklet committee for putting the wonderful booklet together. If someone could share that booklet link in the chat, that would be really great, so that everyone who did not get that booklet can have it and see it. It's really a professional level work. You should, all of the students, you should be putting all of these skills into your resume, because your event planning, your booklet planning, your communication, your slideshow, event planning, project management, those are the skills that that you gain without understanding that you're gaining those important skills while you're working on science. And so there's a million things that there's a million different things that you're all getting without you even realizing that that these are the skills to have. So thank you to all the committee members for putting all of this together. Thank you to our 11th graders who are phenomenal leaders. Thank you to our 10th graders who are learning to step up and to learn from our older students. Thank you to our administrators, to the Billy Noyster Project, to the parents, to the custodians, and to all our loved ones. Have a wonderful summer, everybody. Be safe, be well. Life is good.
Thank you. Have a good one, Mauricio.
You too, Dave.
Have a good one, everyone.
Thank you. Take care, everyone.
Bye.
Be well.
Thank you. It was good. Thank you.
Thanks, everyone. It was really good to see you guys.
Thank you there, Mike. We
Great.
didn't
Yeah,
we didn't
it was
have to
great.
bother you too much. Yeah,
No,
you know,
not a- the one thing I was the message I was trying to get to you was that the chat was disabled, and the MCs were talking about putting questions in the chat.
Oh,
I wasn't able to get
no.
your attention, but I don't think anybody was stressed out about it.
Okay. Yeah. I disabled the chat. That's right, earlier. Mm-hmm. Yeah. Did you get my chat, though? Like stuff that I put on the chat?
Yes. Yeah, yours
you
came
did.
through.
Okay, good. Yeah, folks are probably wondering what was going on. Yeah, I did that just to kind of keep things a little simpler, and because sometimes I think folks start like throwing a whole bunch of stuff on the chat, and then it detracts from the voices.
Yeah, it's nice when people talk.
Yeah.
Raise your hand and talk.
Oh, yeah, and I'm still recording, so let me stop recording this thing. How do I stop recording? Whiteboard recording, stop recording. There we go.